Okay, now we had just finished kind of talking about like some basic anatomical features of the cell membrane. And I'm going to back this up for just a minute just to kind of spend a little bit of time kind of reviewing. So the cell membrane, we're trying to define the purpose and really trying to understand what its role is for the cell. And we said, probably the best thing that it does is stopping the spread of disease or, keeping everything in place and intact. And when we look at the functioning of the body, we realize just how important this is, especially, when we were talking about the mitochondria and cellular respiration. We said number 2, it's going to keep or it's going to get rid of materials. It's going to be able to diffuse things between cells, share ATP, share solutes and different minerals and things that the cell needs. And that, of course, would feed into the whole food thing. And then you had to look up some vocabulary terms. You had to look up osmosis, which we said was the diffusion of water. Isotonic solutions equal, so the water is able to move into and out of the cell freely. In a hypotonic solution, you have more dissolved particles inside, so therefore water is going to flood into the cell, trying to equalize itself. And in hypertonic solutions, we have less dissolved particles inside, and therefore the water that's inside the cell, because there's more of it, will leave the cell. And the cell will shrivel. And there's a fancy word that I do want to write. It's called crenate. And that's what happens when a cell is in hypertonic solution. So think about crenation as being like a dehydration of the cell. And then, of course, we looked at the fluid mosaic model, and that was your warm-up, and you got to watch this. I'm sorry, it was not your warm-up. You guys were looking at diffusion and osmosis, but as part of the video that feeds into and explains what the warm-up was, I'll get it right here in a second. The fluid mosaic model shows us that the membrane is not rigid. It is not rigid. And that it is very fluid, if you will. And that fluidity allows its ability to kind of repair itself, and kind of survive in a way. So you also demonstrated this when you had to use the cutouts to assemble your membrane. So you kind of followed the directions and you labeled all the parts, which is where we were in the last lecture. We broke down the anatomy of our membrane. Of course, it's got this phosphate head, which is polar, and it loves water. So it loves H2O. And then we have these fatty acid chains, which are associated with lipids, and it hates or is fearful of water. The terms that we give these, if you're polar and love water, you are hydrophilic. If you are nonpolar and fearful of water, you are hydrophobic. And then we got to our channel proteins. And we related this back to something previous for the electron transport chain in cellular respiration. So how did all of the hydrogen ions get outside into the intramembrane space of the mitochondria? And of course you said, well, it's pumped through channels. And so your body has these cells. These cells are responsible for maintaining homeostasis. They're responsible for keeping you safe. So how do things get into and get out of the cell? The first topic that I want us to explore, let me fix this terribly white screen, is something called passive transport. Okay, so passive transport. And so if things are passive, do they require energy? So no energy required. So no energy has been required. So let's take a look at some passive transport. It's going to work with, this is of course diffusion. I mean, this is, I hate to sound like a broken record, but let's just go ahead and state the obvious. This is diffusing molecules. There are transport proteins. So look at your model. So transport proteins that allow or act as channels to open up and allow things to move in. And the things move ions and molecules. Okay, like what do you think maybe some ions are? Could be some sodium. You can have some chlorine, maybe some molecules. What do you think about molecules? Do you think it'd be like glucose, maybe? Do you think it could be proteins? Do you think it could be ATP? Yeah, so any of these things that we've kind of talked about in class. All right, so there are types, or I want to call them divisions of passive transport. And the first one, of course, is facilitated diffusion. And so facilitated diffusion is, and I'm going to read the definition here, passive transport of materials across the plasma membrane with the aid of transport proteins. So moving stuff with the help of, we said transport proteins. So let's look back to the model that you guys constructed. You know, can you label or can you find your transport proteins? Do you have something that would allow materials to move into and out? And of course, the answer is yes, you do. I have this, we're going to take like what we call a commercial break at this point. And I have these dots, okay, these dots, and I thought it would be really cool for us to kind of review some elements of facilitated diffusion, but also to kind of take a look at isotonic, hypertonic, and hypotonic solutions. So when you get to this point, I want you to let me know in class, and I can get the materials for you that will allow you to do this, and I can kind of help you to do this. So whenever you get to the point, to this point here, just just go ahead and get my attention and let me know, and I can set you up for this, and kind of go through it if you need help with isotonic and all of that stuff. So we're going to use this time as a way to review terms that we previously learned, but we're also going to use that to help us understand facilitated diffusion.